And just little change in the topic, innate immunity, new frontiers, just in line with your webinar series. So let us go first to what is immunology. In very simple language, immunology, the study of the immune system and the threats perceived by various kinds of hosts. And immune system is the defense system of all living organisms. In other words, I would say that all living organisms have capacity to defend themselves by self, non-self discriminatory power, which is endowed with them, all kinds of species. And in case of vertebrates, there are two arms of immunity or immune system, that is innate and adaptive. And of course, innate and adaptive arms are present only in vertebrates. Invertebrates and lower forms of life have only innate form of immunity. And on the other hand, the perceived threats are in the form of various sort of parasites, say viruses, bacteria, fungi, protozoa, helminths, insects, etc. And in addition, there are environmental injurious substances that too are encountered by the immune system and its defense, and all sorts of non-self threats which reside within or are produced within at different times in the immunologically marked self. By that, I mean immunologically marked self means we learn to discriminate self from non-self during our embryonic and fetal life. Okay, then what is the role of vertebrate immune system or immune system in general? Normal immune system functions are defense against harmful foreign substances, that includes environmental substances, homeostasis to remove tissue debris, immunosurveillance to remove mutant cells as they arise in different time during the life, and preservation of identity, that is rejection of allo and xenografts. So all of these are taken care of by the immune system. But as we also know that there are aberrations or dysregulation of the immune functions that could lead to allergies and hypersensitivities, autoimmune diseases, neoplasms and malignancies due to mutations in the immune cells, and immunodeficiencies that could be congenital or due to some suppression or destruction of the immune cells. Now, the types of immunity, they have often been classified as into three types: barrier defenses, innate immunity, and adaptive immunity. Sometimes barrier defenses are taken together with the innate immunity. But barrier defenses are also, say, important, and they are of various sorts: anatomical, physiological, biochemical, and normal microflora. That is becoming very important nowadays, the understanding of microflora interacting with the host, because they have been found to be involved in development of the immune system, both innate and adaptive. And in addition, many other phenotypes of the host are influenced or determined by the microflora that we have in us. And then innate immunity, you can further divide it into humoral and cellular. Likewise, in adaptive immunity, humoral and cellular, but the components and their functioning differs in many respects that I'll come to present in a few minutes. Say, humoral response, humoral components are complement, acute phase proteins, cytokines, even natural antibodies are considered as part of the innate immunity, which are in the mucosal secretions, etc. And then cellular components are various sorts of phagocytes. In addition, NK cells, natural killer cells, mast cells, eosinophils, gamma delta T cells, and say NKT cells also. But the adaptive immunity humoral component is antibody mediated and cellular component is T cell mediated. Antibody mediated component is contributed by the B cells that produce and secrete antibodies later as a result of immune responses. Now, just I have taken this from a recent webinar series. You can see the innate and adaptive immune system cells. There are mast cells, dendritic cells, macrophages, natural killer cells, and complement proteins as molecules, and then neutrophils, eosinophils, basophils as granulocytes. In addition, even if they are lymphocytes, say gamma delta T cells, on which I had opportunity to work during my postdoc program at Indian, say, Institute for Animal Health in UK, and they behave as both innate and adaptive immune cells. Likewise, there are natural killer T cells which phenotypically share the markers of natural killer cells and T cells, and they are involved in immunoregulation some activities. Say, and then importantly, there are two B cells and T cells. T cells are further divided into broad categories: CD4 positive T helper cells, CD8 positive T cytotoxic cells or CTLs. B cells, you know, produce antibodies which mediate the defense. T cells, through their activation to become effector cells, they carry out effector functions. The CTLs will kill the target cells which are infected with viruses or intracellular pathogens, or say effector T helper cells, they effect through cytokine release and various mechanisms. Okay, so the classical components of innate immunity are phagocytes. Macrophages and neutrophils, you know, neutrophils are the first to come. They are very vigorous, short-lived phagocytosis, but often they succeed in preventing infections without even our knowing that, because we know that daily we encounter so many threats, but we don't suffer from diseases, infectious diseases so often. And that means someone is working for us, and that someone component is phagocytes. Of course, innate immune molecules, barrier defenses, all of these contribute to those defenses. And this is a picture taken from QBs, neutrophils and macrophages. They differ in morphology and in some sort their functions also. They macrophages come later in, say, infection, during sustained, say, infection. And this is a picture showing the bacteria, how the macrophages extend their processes to catch the bacteria, engulf and destroy them. So this is the classical viewpoint of, say, phagocytes as components of the innate immunity. Now, we should know that innate immunity is indispensable for host defense. In fact, this we come to know in cases where there are congenital deficiencies. Congenital deficiencies of innate molecules such as complement components result in recurrent pyogenic infections. They make individuals' life hell because of these problems. Likewise, there are congenital defects in phagocytic function that leads to delayed wound healing, recurrent infections, stunted growth, etc. So that shows, their deficiency shows they are indispensable for host defense. Now, having said that, we would like to compare the innate immune system with the adaptive immune system. In what respects these two sorts of immunity differ? And the point of difference, if you see the recognition system, In case of innate immunity, pattern recognition receptors are present on the cell surfaces of dendritic cells, macrophages, and other cells, which recognize the conserved molecular patterns on pathogens. This sort of molecular explanation has been forwarded only during past 2 decades or so. Adaptive immunity, we know for long that there are BCR, TCRs, and secreted antibodies which recognize specific chemical configurations in the form of epitopes on antigens. So this is a basic difference between two recognition systems. Manner of development, germline. So innate immunity is germline encoded. That means all the genes are present and they will encode all kinds of molecules and cells. That means it is determined at species level and there are no clones existing, unlike the adaptive immune system clones, say, that is B cell clones, T cell clones, making the whole huge repertoire. So somatically shaped gene rearrangement of BCR and TCR occurs in the primary lymphoid organs, that is bone marrow or bursa fabriceus and thymus. And then extent of diversity is, now you can say that innate immunity has discriminate self from non-self. That means, but that power is limited in the form of only limited number of conserved molecules. Whereas in case of immunoglobulins, paratopes, there are, say, huge number of paratopes for TCR and BCR receptors. Then immunological tolerance is not applicable. Tolerance to the self is achieved by clonal deletion and clonal energy in case of adaptive immune system. Operational level is, we know that this is first line of defense and this appears later, the adaptive. Say phagocytes are the cellular immunity component, NK cells and some other cells, say gamma delta. Here it is effector T lymphocyte subsets. Molecular immunity elements are, as we have already said, complement, cytokines, acute phase proteins. Here it is antibodies, they are secreted forms. Say memory is a remarkable difference in the classical view of innate immunity is memory is absent. We have been taught since our student days that memory in the innate immunity is absent, and which I will come later talking about the new concepts that are emerging and that show some form of immunity or innate immune memory which is being currently investigated. Memory, of course, is present in the form of memory B and T cells in adaptive immunity. Secondary immune response is because the innate immunity behaves as in the classical viewpoint, always unchanged because of lack of memory. In case of adaptive immunity, there is higher magnitude and different quality due to memory in the secondary immune response or anamnestic immune response. And the cognitive activation and effector phase, you can divide the immune response, innate as well as adaptive, in 3 phases, that is cognitive, followed by activation, followed by effector phase. Although it is not very well defined or molecularly explained in case of innate, but whatever happens within a few minutes, whereas it takes a few days time in case of adaptive immunity to develop. And this immunity innate is found in vertebrates and invertebrates, all forms, whereas this is exclusively present in vertebrates. Now, having said that, we have seen the classical point of innate immunity. The paradigms of innate immunity are changing, and they have changed during last 2 decades or so. I'll come to discuss these in brief, one by one. But let us see this table, that what we say that self-known self-discrimination. which is present in both forms of immunity. Only thing is that molecular explanations for innate immunity have been forwarded only during past 2 decades or so. After the discovery of pattern recognition receptors interacting with pathogen-associated molecular patterns or microbe-associated or danger-associated, which may arise within the body. So, and these explanations have been forwarded, but this discrimination is different in form, different from the adaptive immunity. Then comes the specificity. Specificity and memory are the hallmarks of adaptive immunity. We know that. But these features have been explored in innate immunity also recently. And we'll come to know that specificity in case of innate immunity can be said to be broadly specific, that phagocytes can differentiate, they won't engulf our self tissues unless there is some damage to it, but they will efficiently engulf the foreign substance as it happens. But its molecular explanations, the specificity molecular explanation have been forwarded recently. And these are, say, which molecules are involved in that? Say PRR, there are pattern recognition receptors, first of which were toll-like receptors discovered, and later NLR and say RLRs and some others, they have been discovered. They interact because there are different kinds of molecular patterns. Say, for example, LPS is one, CpG-DNA or muramyl dipeptide and many others. They are recognized by different receptors. That means the specificity for LPS is different from that of specificity for, say, CpG molecules. So in that way, a broad specificity resides, and there are molecular explanations today available. And there are signaling pathways, transomics, which describe the different states of phagocytosis and the link between adaptive and innate immunity. And then memory. A new concept known as trained immunity has been described and gaining strength nowadays. And that trained immunity in innate immune system has been found, and that is achieved through epigenetic, transcriptional, and metabolic reprogramming, and which has been, say, found to have impact on vaccinations and can also have impact on therapeutic interventions. Now, another important aspect which relates to innate but also to adaptive immunity is the microbes, microbiomes. Microbiomes are the whole, say, entire community of microbes which reside in our gut, respiratory tract, skin, etc., different niches. And it is now believed that microbiomes co-evolved with the host species, and a concept known as holobiont or phylosymbiosis concept is being forwarded. By holobiont concept, we mean that our microbes in our species, the Homo sapiens and other species, they are intimately intertwined with us, and they control or regulate various phenotypes of the host. On the other hand, host also controls which kind of microbes will reside in us. And this is how they have co-evolved to suit to each other and to give survival advantage to the species. And this concept is gaining strength nowadays. And another point is host microbe environment interactions. They determine host phenotypes such as immune responses, both innate and adaptive. And then metabolic traits, behavior, and also connected to nervous system. So the host determines, on the other hand, microbiome composition. And they have impact on vaccination also. Now, in short, that the innate immunity first thing that we were talking of specificity and how the innate immunity recognizes the pathogen through TLRs or other PRRs, which will say induce signaling pathways and which are say signal transduction goes to induce new say transcription of the cytokines, set of cytokines, co-stimulatory molecules, and antigen presentation enhancement through MHC class 1, class 2. So this is the bridge between adaptive immune cells and the classical innate immune cell. That is the antigen present in that form, antigen presenting cell. So there is a link between innate immunity and adaptive immunity, and without which you cannot think of T-dependent immune responses. And this, the microenvironment created by these cytokines, in addition to these interactions, will determine which path the naive T cell will differentiate into further, say TH1, TH2, TH17, TH19. Now there are many different subsets of the T cells known, and which will produce their signature cytokines. They will determine the outcome of the immune response, whether it is going to be cell-mediated predominantly or antibody-mediated or a mixed type of immune response against many different kinds of, say, extracellular bacteria, helminths, or viruses, or say this way. So these things, molecular explanations are being revealed, have been revealed to a large extent. Now, and we have told that there are many different kinds of PAMs, which the source is bacteria or say fungi, viruses. So these PAMs, molecular patterns, they reside on many different kinds of infectious agents, and they are sensed by TLR. And now we know that NLRs, RLRs, CLRs, etc. Okay, then another point, when we were saying microbiota affect both innate immune system and adaptive immune system development and functions. There is a recent article readable that is Zhang et al. In Cell Research, and I have taken information from there. And this says that there is interplay between commensal microbiota and the mammalian immune system development and function. Then microbiome has role in training and development of major components of host immune systems. And the immune system orchestrates the maintenance of key features of the symbiosis. So microbial symbiosis is a result of host microbe interaction, and which is mediated by immune system. Often the commensals, the immune system does not respond in an active form. That means it just immunoregulates, causes immunotolerance to the commensals, but immune response, productive immune response to the pathogens. So this fine tuning of the immune system is remarkable. And then if there occurs imbalance in the microbiota immunity interactions, that leads to pathogenesis of a multitude of immune-mediated disorders. Some are, say, of the gut-associated inflammatory conditions, but it's beyond gut also, so that can be affected. So microbiota affect immune functions and vaccination outcome. There is a very interesting article recently, last year, by Pulendran, who is a well-known vaccinologist, and they say that disturbed microbiota can affect adversely the vaccination outcomes. In addition, you will find several articles recently that say that dysbiosis results in inflammatory diseases and immune response is involved or dysregulated in such situations. So these biases can affect immune interventions and vaccine outcome. So this is important to note, particularly in this COVID pandemic. Next, so immune microbiota interactions, as I said, can affect not only the gut or local systems, but also gut-liver axis, gut-lung axis, and gut-brain axis. So, say short-chain fatty acids, LPS, MAMS, probiotics, Klebsiella pneumoniae, and some probiotics here, including short-chain fatty acids. So they can affect in various ways, and all the kinds of axes here, affecting many different systems. Then the innate immunity, now coming to a very important topic which has interested me and to share with you is the innate immunity can be trained, which is not very digestible concept for the classical immunologists of the 80s and 90s. But now the things are changing, and there is convincing scientific evidence being produced, and very recent articles like this one in this year, and another one also in the same, say, June science issue. And I'll just tell about their findings that for past 1 decade or so, the work has been going, and now there is sufficient evidence that supports that there exists memory in the innate immune system. So monocytes and NK cells exhibit memory, and that memory is through epigenetic and metabolic reprogramming of these cells. In the next slide, I'll tell in a picture that may be understood better. And then monocyte macrophages acquire alloantigen specific memory to induce organ transplant rejection in mice. That's very convincing, evidence they have produced. That MHC1 specific, this is allo, means donors MHC memory by paired Ig-like receptor PIRA on macrophages. On macrophages occurs this receptor, which is a member of the Ig superfamily, and such molecules have been found in invertebrates also, like DSCAM in insects, fibrogen-related proteins in mollusks, scavenger. That means we know that invertebrates do not have adaptive immunity, but they do have innate immunity, and innate immunity memory has been shown, or it is adaptive like memory has been shown in insects and say other lower forms of life, invertebrates. So, and this situation in mouse is like that. And then increased, that means what is trained immunity impact? That increased responsiveness by trained immunity against certain infections or vaccinations can induce protection against specific and non-specific infection. We'll give a very recent example, which was also talked about in the COVID control in the next slides. Now let us see, there are different forms of innate immunity. Memory. Say, how it can be explained is, we know that the monocyte macrophages in the resting state, if they get first stimulus in the form of infection, they get activated, and there is a release of cytokines as shown in some previous slide. Now, from this state, activation state, there leads to development of memory, and which can be divided in two kinds. One is through the epigenetic changes, the memory that develops is known as trained immunity. In the other way, when this memory is mediated through PIRA receptor on macrophages, this becomes MHC1 specific response memory, and this is specific innate memory. So trained immunity and specific memory both have been demonstrated, at least in mouse, and have been suggested for other systems. Now, another important article was whether BCG vaccination can protect against SARS-CoV-2 virus infection. And the original authors of this, they were working for some past few years. They found that BCG vaccination reprograms monocytes through epigenetic changes or controls. And this leads to production of inflammatory cytokines like IL-1 beta, interferon, and necrosis factor, IL-6 during subsequent viral infections. And this has been demonstrated that BCG vaccinated individuals can be protected against respiratory syncytial virus, influenza virus, HSV2. These are findings of the well-conducted trials after BCG vaccine in humans. Now they were just suggesting that it is possible that often the respiratory infections by viruses can be protected or can be taken care of in BCG vaccinated individuals. So it is possible, they said that it is possible that SARS co-infections, particularly the mild and moderate cases, can be taken care of in BCG vaccinated individuals. And they, although the data is still not very foolproof, they said, but it is suggested that, or it seems that those who have been BCG vaccinated, particularly children, they are more protected against the severe infections. And how this innate immune memory is achieved, say here in the graph, that primary challenge vaccine or infection will give a first response, and on second infection or encounter, there is a trained response which is higher in magnitude and longer lasting. So being longer lasting, higher in magnitude, it is said to be a trained immunity trait or a memory specific or memory-like response. Now, next, in the same article by O'Neill and Natia, they in this year, June, they found that how it is achieved, this BCG-induced trained immunity, that it is through pattern recognition receptors interacting with the PAMPs from the BCG. They will do metabolic reprogramming. Several different epigenetic enzymes are affected by metabolites, which are cofactors for enzymes, and they do histone modification. So at that level, say in case of primary response, the chromatin is a closed, enclosed, shape, which causes low response, whereas in case of reprogrammed in the secondary response, chromatin becomes open, and that means it allows more transcription of those, say, genes which are involved in, say, pro-inflammatory cytokine production, and this is how they achieve. Now, what they proposed in this is, now, COVID pandemic is a situation where we don't have a specific therapeutic agent and a vaccine. Both are lacking. So in that situation, they said that if there is emergence of a new pathogen, you can use the trained immunity inducers during this phase so that there is a broadly specific immunity provided for a few months' time. Now, after this, so that means you are buying time by adopting such approaches of broadly specific immunity. And so that by in a next few months' time, you are able to develop and use the specific vaccine. This is how it is proposed. It seems attractive. But the COVID situation is now beyond that. It has passed that stage. Now, trained immunity versus adaptive immunity, they say that it is just the table that I have shown previously, it is similar to that, that specificity inducers of trained immunity are pathogens and derived PAMPs. Here, adaptive immunity, antigen presentation. Receptors are PRRs. Here it is TCR and secretory immunoglobulins or BCR, and with co-stimulation and cytokine. Clonality is absent, as we have said earlier. Cells, you know, they are of the trained immunity involved monocyte, macrophage, NK cell, DCs. ILCs are which one, I don't know, I've forgotten this. B and T lymphocytes, they are other innate immune cells. And then memory. But here, just we need to remember that trained immunity is has memory which can be only lasting for months, whereas adaptive immunity can last for years and several years, even lifelong. So memory mechanism here is epigenetic modifications. Here it is clonal expansion and differentiation of memory cells, specific memory cells. Now there are examples of pathogen-associated molecules with experimental evidence of cross-protection. So this is an example of trained immunity-based vaccines. Say LPS, which is part of the gram-negative bacteria, can cross-protect against Staph aureus. This has been demonstrated experimentally. Likewise, peptidoglycan component can work against Toxoplasma. Flagellin of gram-negative bacteria can work against gram-positive and rotaviruses. And this is Fimbri H of E. coli against influenza virus. Beta-glucan, chitin, and CpG-DNA, they all can cross-protect. That has been shown. Now, based on these observations, it is suggested that trained immunity-based immunostimulants can be used as, say, adaptive immune, specific immune response against bystander pathogens. And this is the way or the path to achieve that. Likewise, trained immunity-based adjuvants, if you select adjuvants for vaccines, particular antigens, these will train the immunity, innate immunity. These will also be enhancing immune response against antigen X, but also after training innate immunity, also antigen Y adaptive immunity can be achieved. This is how they propose. Now, the aim of this presentation was just to bring before you the new frontiers which are being, say, which investigated, and the new knowledge is being added in innate immune responses, which is relevant to be applied in vaccination programs and in therapeutic interventions and say these areas. So just to conclude, and innate immune system is evolutionarily more ancient than adaptive immune system. Innate immune system molecules and cells are the frontline warriors for protecting the host. That must be clear. And innate immune system links to adaptive immune system in vertebrates and initiates adaptive immunity. That too is indispensable. New concepts in innate immunity are emerging, particularly host microbiome interactions, which are affecting both innate and adaptive immunity. And they can affect vaccination outcomes, because in development of vaccines, there is a stage where we challenge with the pathogen after the trials are successful. And the challenge studies often are not in the host proper in case of humans, they find animal models. But the question is, the microbiomes of the animals are different from microbiomes of the human. So the results might be misinterpreted, and these apprehensions have been raised from microbiome interactions point of view. And then trained immunity is a form of specific memory in innate immune cells. This is being emerging and gaining strength. And with further experiments to strengthen this concept, we'll find that trained immunity can be exploited to formulate vaccines, to have desired outcomes, and likewise therapeutic interventions. Now, I would like to express my gratitude to the Institute for Animal Health, Purbright, where I did postdoc research in immunology, particularly on gamma delta T cell biology, showing its innate-like characters. Then, of course, ICR for my current emeritus scientist scheme, which is being run or implemented here in LUAS, which is my workplace for research. So, and thank you all for patient listening.